Hi, I'm Jing-Qiong. I also go by Katie. I'm an associate professor of neurology at the Vanderbilt University Medical Center.
I'm Sarah Poliquin. I'm a 5th-year graduate student here at Vanderbilt.
And hi, I'm Gerald Wasu, a 3rd-year graduate student here in the lab with Jing-Qiong.
Hi, I'm Carson Flam. I'm a 2nd-year undergraduate student here working in a lab with Dr. Jing-Qiong.
This is for a video abstract of our recent paper published in GABRB3, August 19. So this paper is a part of our serious work on understanding pathophysiology of genetic epilepsy associated with GABAA receptor mutation and more recently with GABA transporter mutation. This paper has focused on GABAA receptor GABRB3 mutation. Our research aims to understand what's going on in the patient's brain if they carry a mutation. These mutations are associated with a wide spectrum of epilepsy syndromes. GABAA receptor is a pentamer composed of 2 alpha, 2 beta, 1 gamma, or delta. Mutations in different subunits are often associated with different epilepsy syndromes. For example, mutation in GABRB3 associated with febrile seizures, generalized epilepsy with febrile plus or more severe kind of epilepsy called Dravet syndrome. Interestingly, mutation in GABRB3 associated with childhood absence epilepsy, infantile spasm, and Lennox-Gastaut syndrome. But 10 years ago, we have reported mutation in the same gene, GABRB3, associated with autism with maternal transmission. Now we are reporting multiple mutations in the same gene, GABRB3, associated with variable epilepsy syndromes, unexpected sudden death in epilepsy, autism, and intellectual disability. In this paper, we first compared with multiple mutations, and now we focused on 2 mutations we call the mutation A and the mutation B. Mutation A is associated with a more severe kind of epilepsy called Lennox-Gastaut syndrome. The patient also has a neurodevelopmental delay, while mutation B, a patient associated with mild epilepsy called juvenile absence epilepsy. The patient has normal cognition. So that's quite interesting. We wonder why the mutation in the same gene give different epilepsy severity. So we first test the human recombinant GABAA receptor with the mutation. We tested in cell models, including neuron, and then we tested in the mouse model with GABRB3 deficiency.
This schematic shows the postulated mechanisms of the 2 mutations we're studying. In subunits containing mutation A, there is decreased trafficking of the subunit to the synapse, resulting in fewer receptors. While in mutation B, the subunits are able to traffic to the synapse, however, the receptors have impaired function. Both mutations, however, result in altered partnering with the gamma-2 subunit that binds the receptors to the synapse.
Based on the findings from cell and mouse models and from the patient history, we conclude the intrinsic property of those mutations in GABRB3 can give rise to epilepsy syndromes with different severity. In addition to GABRB3, other genes also give a similar clinical phenotype.
Although there are other genes associated with the Lennox-Gastaut syndrome phenotype, including SCN1A, SLC6A1, and KCNT1, we are continuing our work on GABRB3 by investigating in our mouse models a rescue phenotype with genetic and pharmacological approaches.
We are very grateful to all the previous trainees, postdocs, and students who are not here anymore, and all the collaborators who have made a wonderful contribution to this work. If you want to learn more about this study, please read the whole paper in Brain Online.